Dear students, in previous video we have understood restriction enzyme. What is the function of restriction enzyme? To cut DNA, right? To cut DNA. Next step that we are going to understand is separation and isolation of DNA fragments. Look, by the action of restriction enzyme क्या हुआ है? DNA के हमने छोटे छोटे टुकड़े किए हैं. Yes or no? Yes. How we are going to separate these DNA fragments? By a technique called as gel electrophoresis, and today we are going to discuss this technique. So what is gel electrophoresis? It is nothing but separation and isolation of DNA fragments. These DNA fragments are created by action of our restriction endonucleases. We very well know that. Now let us understand what is this electrophoresis. Once again वही लिखा है मैंने. After cutting of DNA by restriction enzyme, fragments of DNA are formed. Now these fragments of DNA can be separated depending upon their size and length by a technique called as electrophoresis. This technique was developed by A. Tiselius in the year 1937. So look what I have written. Separation of DNA fragments according to, most important thing is here, according to size or length, okay, is done by a technique called gel electrophoresis. किसने डेवलप करी थी ये टेक्निक? A. Tiselius in the year 1937. Now as such, this electrophoresis can be employed for separation of not only DNA. This can be employed for separation of your RNA as well as proteins also. So वापस से मैंने क्या लिखा है? Electrophoresis is a technique of separation of DNA, RNA or proteins on the basis of size under influence of electric field. बहुत important चीज, okay? This machine operates in presence of electric field. So under influence of electric field, right? So that they migrate in direction of electrode bearing opposite charge. Simple as that. Say for example, if you are talking about our DNA, तो DNA का क्या charge होता है? DNA are negatively charged. So under electrical influence, they are going to move towards positive electron. They are going to move towards opposite electrode. Am I clear? So once again क्या लिखा है मैंने? They are going to migrate in direction of electron bearing opposite charge. Simple as that. DNA का charge negative. This negatively charged are going to move in direction of electrode bearing opposite charge. मतलब कौन सा charge होगा electrode का? Positive charge. That is, we are talking of our anode. This is your electrolytical cell. Okay. So लिखा है मैंने, that is positively charged molecule. अगर आपका molecule positively charged है, it is going to move towards negative electron. If your molecule is negatively charged, say your DNA, then DNA is going to move towards positive electron. Medium भी तो होना चाहिए by which they can pass, yes or no? So of course there will be a presence of a medium in form of gel matrix. Okay, so once again what I have written, this molecules are going to move towards electron bearing opposite charge through a medium, or you can say it as your matrix, or you can say it as your matrix. So here if you are talking about your electrophoresis, it is performed in gel matrix and thus it is called as gel electrophoresis. इसकी वजह से इसको क्या कहा जाता है? Gel electrophoresis. Now which gel we are going to use? As a matrix we are going to use agarose gel. We are going to use agarose gel, which is basically a polysaccharide. जो क्या है? Polysaccharide that is your carbohydrate. That is your carbohydrate, and it is extracted from seaweeds. It is extracted from seaweeds. Okay. So when your DNA fragment move through this pores of agarose gel, our DNA fragments are separated on the basis of size or length. Simple सी बात है. Now let's talk about agarose gel. How this gel is formed. So we have to dissolve this agarose in hot water. Once you will dissolve in hot water, then you have to allow that solution to cool. Once the solution is cooled, there will be formation of double helix like this. There will be formation of double helix like this. Okay, so what I have written, agarose dissolve in water. When this solution cools, double helix form and become arranged laterally. This all double helix arrange laterally. Can you see this? Yes. This results in formation of thick filament. What is this? This results in formation of thick filament. ठीक है, कूल करा आपने, डबल हेलिक्स फॉर्म हुई. दिस डबल हेलिक्स अरेंज्ड laterally to form thick filament. Okay, and these filaments are cross-linked. ऐसी एक थिक फिलामेंट मैंने यहाँ पे ड्रॉ करी है, ऐसी बहुत सारी थिक फिलामेंट फॉर्म होगी. एंड ऑल दिस थिक फिलामेंट्स आर क्रॉस लिंक्ड, जिसकी वजह से देयर विल बी फॉर्मेशन ऑफ पोर्स. ड्यू टू दिस क्रॉस लिंकिंग there is formation of pore. Okay, that is formation of pore. And of course this pore size again depends on concentration of agarose. किस पे डिपेंड करता है? Your pore size depends on agarose concentration. Am I clear? So this is for your representation. Okay, this is your gel electrophoresis instrument in which you are going to perform this separation technique. Can you see this? 1, 2, 3 and 4. Blue color one. 1, 2, 3 and 4. These are called as wells. Wells are nothing but in this you are going to add your DNA solution. इसमें आप क्या करने वाले हो बेटा जी? You are going to add your DNA solution. Over here that is your matrix. That is your agarose gel. Okay, and this is under influence of electric field. Simple as that, right? What is the principle behind this? Smaller the fragment, जितना छोटा fragment होगा उतना ही वो दूर चला जा रहा है. Can you see this? Smaller the fragment, they move farther. वापस से एक बार, जितना छोटा fragment होगा it will go further. Okay, so these are your DNA bands. Can you see this? These are your DNA bands. This is the larger DNA band. These are your larger DNA band, whereas these are your smaller DNA bands. Okay, as such, this separated DNA fragments will be visible only after staining this DNA with ethidium bromide. ETBR. This is the abbreviation. Ethidium bromide. Okay, this DNA fragments you have to stain with your ethidium bromide, followed by exposure of UV rays. Followed by exposure of UV rays. Only then you are able to see orange color bands. You are able to see this orange color bands. Okay, now what you have to do? From this gel you are going to separate this fragment. Simple as that. From this gel you are going to separate this fragment, and this process is called as elution. ये प्रोसेस को क्या कहा जाता है? इल्यूशन. देखो क्या लिखा है मैंने? The bands are cut and extracted from gel piece. Okay, यहाँ से आप कट करो, फिर जेल में से उसको एक्सट्रैक्ट करो. ये प्रोसेस को कहा जाता है इल्यूशन. Once you people have separated DNA fragment, desired DNA fragment, what you are going to do? You are going to ligate with vector. Right, you are going to ligate this gene, this DNA fragment with vector for creating recombinant DNA, for formation of our recombinant DNA. तो ये क्या हो गया आपका gel electrophoresis. What are the key points? Electrophoresis works on the principle of what? Size, length, and of course electric field. Okay, molecule move towards the electrode of opposite charge. Specifically we are talking about DNA. DNA is negatively charged, so this DNA move towards your positively charged electrode. Simple as that. Okay, so it runs along your gel, agarose gel, but this separated DNA fragments are only visible if you are going to stain DNA with ethidium bromide followed by exposure with UV rays. And lastly, your elution technique. क्या होती है elution technique? Once the process is done, you are going to cut this DNA, you are going to remove gel, and ये process को कहा जाता है elution, followed by which the desired piece you are going to ligate with vector for creating your recombinant DNA.
In this video, I will be discussing how gel electrophoresis is used to separate DNA fragments of different sizes. So let's say you have a sample that has a bunch of copies of this piece of DNA, and you use a restriction enzyme to digest this molecule into two pieces. Here is the restriction site, so we cut here and create a small fragment of DNA and a larger fragment of DNA. Now I want to be able to isolate this fragment to use later in my lab. Right now, I just have a sample containing some medium-sized pieces mixed in with some smaller-sized pieces of DNA. I can use gel electrophoresis to separate all of these medium-sized molecules from all of these small molecules. How do I do that? So this procedure uses a gel, usually made of agarose, that is placed in a buffer that has ions that can carry current. The gel contains indentations called wells, where we inject the DNA sample that we want to separate. We then apply an electric field that has a negative pole on the side of the gel with the wells and a positive pole on the other side of the gel. We should remember that DNA is negatively charged due to the phosphate groups in its backbone. When the electric field is applied, the current is running through the buffer. The negatively charged DNA fragments move away from the negative pole toward the positive pole through the agarose gel. In this process, the larger the DNA fragment, the slower it moves, causing the fragments to separate based on their size as they travel. In this case, let's run two samples through our gel. In the first well, let's run an undigested sample that only contains the full large segment of DNA that has not been cut by the restriction enzyme. In the second well, let's run the sample that has been digested by the restriction enzyme, resulting in this medium-sized fragment and a smaller fragment. As the gel runs, the smaller fragment will move faster through the gel than the larger fragment, and we end up with something that looks like this. In the first lane, we can see that our large undigested molecules do not move very far in the gel. In the second lane, we can clearly see two separate bands of DNA. We have successfully separated these two different sized fragments, the medium here and the small here. And we can cut either one of these fragments out of the gel to use the DNA in further lab protocols, perhaps by inserting it into a plasmid and transforming some bacterial cells. So that is gel electrophoresis. We load DNA fragments of different lengths into a gel, and then the smaller ones move more quickly than the larger ones when current is applied. If you want to learn more about how you can digest DNA using restriction enzymes, see my video on restriction enzymes. If you're interested in how you can insert isolated DNA fragments into a plasmid and then transform bacterial cells, see my video on transformation.